We now move in to our amino acid derivative discussion. Many important compounds within the body are going to be derived from amino acids. So as you can see here, phenylalanine and tyrosine are two amino acids that are going to be precursors to the synthesis of our catecholamines such as dopamine, norepinephrine, and epinephrine. Now thyroxine is going to be our thyroid hormone, and it's going to be derived from tyrosine as well, while our pigment melanin is going to be derived from Dopa. Now we've got another amino acid that we need to take a look at here, and that's going to be tryptophan. Tryptophan is going to be used to make niacin and serotonin. Now when tryptophan is going to be used to make niacin, we need to use the vitamin B6, which is pyridoxine. Now, as a quick aside, how many ATP does an NADH make when it exchanges its electrons for the electron transport chain? That's right, that's going to create 3 ATP equivalents. Remember we talked about that during oxidative phosphorylation. Now, using vitamin B6 and tetrahydrobiopterin, so BH4, tryptophan can be used to make serotonin. Now, this tetrahydrobiopterin is used to transfer a hydroxyl group to a substrate, tryptophan, and it can also be converted to the serotonin here, which in turn can be converted to melatonin, which is important for sleep. Alright, this is why people associate eating turkey on Thanksgiving with being tired after the meal, since turkeys have a lot of tryptophan in them. Then also, we've got histidine. Now, vitamin B6 is used here as well, so this pyridoxine is used all over the place. And it's going to be used to convert the histidine to a histamine, which is important in the allergic response. A lot of you with allergies curse histamine every day as the bane of all of your problems. Now I've got a couple more that we need to discuss here. This glycine is used to make heme. So now we need to sidetrack to the heme synthesis for a second. A third use of vitamin B6 is in the first step of heme synthesis, which is the conversion of a glycine to a porphyrin. Now with that said, can you think of why a B6 deficiency can lead to a sideroblastic anemia? B6 is needed for this step. Why would a B6 deficiency cause a sideroblastic anemia? Glycine is combined with succinyl-CoA by the enzyme allosynthetase, which is used to make the delta-aminolevulinic acid in the rate-limiting step of heme synthesis. So this is the rate-limiting step combining our reactants to make an intermediate here. Now this is why a deficiency in pyridoxine can cause sideroblastic anemia. Sideroblastic anemia is covered more in detail in the heme-onc section, but simply it's a defect in the heme synthesis that causes ringed sideroblasts. So if we bring a picture in here, we can take a look. You can see in the image that this erythrocyte is loaded with iron stuck in the mitochondria, creating a sideroblast. Alright, so now let's go back to our discussion on amino acid derivatives. In heme synthesis, you need the amino acid glycine and B6 to create our heme eventually. Now, the amino acid arginine can be used to create creatine, urea, and nitric oxide. And then lastly, it can have the amino acid glutamate to create GABA and glutathione. Now, GABA is going to be an inhibitory neurotransmitter, and it's going to use the vitamin B6 as well as a cofactor. And it can also be used to make the glutathione, like I said, which is an antioxidant, which we already briefly talked about earlier. And this needs NADPH to be regenerated. So, we have the same pathway that we just saw in the amino acid derivative slide. It's a pretty basic pathway, so now let's fill in this picture just a little bit. The catecholamines are the hormones derived from phenylalanine, which include dopamine, norepinephrine, and epinephrine. The first step in the catecholamine synthesis is going to be the conversion of phenylalanine to tyrosine via the enzyme phenylalanine hydroxylase. Alright, so let's blow this up just a little bit. This step requires tetrahydrobiopterin as a cofactor, which reduces it to dihydrobiopterin. So, in our reaction here, as the forward reaction occurs, we take a tetrahydrobiopterin and convert it into a dihydrobiopterin. Now, dihydrobiopterin reductase is going to be used to regenerate our tetrahydrobiopterin. Now, it's used along with NADPH here to restore this dihydrobiopterin into a usable form of tetrahydrobiopterin using the NADPH in the process. Now, speaking of NADPH, here we go. What disease results from a decrease in NADPH synthesis? Now, it's an all-important board question, so hopefully you're remembering it's G6PD, where the enzyme glucose-6-phosphate dehydrogenase is defective or deficient. And can you tell me what you might expect to see on a peripheral smear of such patient with G6PD? Remember, you need to see those bite cells on a peripheral smear of the bone marrow, which represents partial red blood cell destruction by phagocytes. The second step is going to convert our tyrosine to a dihydroxyphenylalanine, or Dopa for short, which requires the enzyme tyrosine hydroxylase, and again uses the tetrahydrobiopterin as a cofactor. Now, here's our reaction just zoomed in a little bit. This step is very similar to the step that we just saw, with the different well-named enzyme in the process. Alright, so again we see the same tetrahydrobiopterin being used, and we need to use the dihydrobiopterin reductase along with an NADPH to reconvert it. Now in the third step, the Dopa is converted to DOPAmine. Now this requires vitamin B6 and dopamine decarboxylase. So as you can see here, I just zoomed in on the reaction a little bit. We see our Dopa decarboxylase. We've got our cofactor, our pyridoxine, which is showing up all over the place here. This step is going to be inhibited by carbidopa to treat Parkinson's disease. So Parkinson's disease. Alright. As you'll see in the neurology section, Parkinson's disease is caused by having too little dopamine. Do you know why a drug that inhibits dopamine synthesis actually helps a patient with Parkinson's here? That's because it's only going to inhibit this reaction outside of the blood-brain barrier, so more likely in the periphery. Here, we see carbidopa, which is going to inhibit this step. However, it's going to only occur in the periphery. Alright, and this is going to allow more Dopa to be available in the brain, which can be converted to dopamine within the brain. So we're going to limit this reaction in the periphery, so more Dopa will be available in the central, in the brain, where it can be then converted to dopamine. Because remember, Parkinson's is just a simple deficiency of dopamine. You'll learn more about that in the neurology section. Now this way, there will be a higher dopamine level in the brain. We just tied in a clinical correlation to our science, to our background science, our biochemical metabolism. Now, we need to make those connections for the boards because that's how they'll test this material. Alright, back to our catecholamine synthesis pathway. Dopamine is then converted to norepinephrine by the enzyme dopamine beta hydroxylase. So I zoomed in on this reaction here. And this is going to require vitamin C as a cofactor. Do you remember what pirate collagen disease results from a deficiency of vitamin C here? Yeah, that's right, scurvy. We talked about scurvy earlier. Do you remember how scurvy presents? It's going to present with impaired wound healing, swollen gums, and also bruising, corkscrew hair, perifollicular and subperiosteal hemorrhages, and then hemarthroses. So, this is my favorite pirate disease because pirates that lack vitamin C, well, out on the sea. Now, what's a pirate's favorite letter? Well, you may think it's R, but their true love be the C. Alright, so in summary of all this, vitamin C is used in the catecholamine synthesis pathway, where it is used as a cofactor to convert dopamine into norepinephrine using the enzyme dopamine beta hydroxylase. In the final step here, norepinephrine is going to be converted to epinephrine by the enzyme phenylalanine N-methyltransferase. Big long word here. Now, since this reaction adds a methyl group to norepinephrine, it should not be surprising that you'll see SAM, the methyl transfer man. Remember, I talked about that in our activated carriers discussion. It's going to be acting as a cofactor to donate that methyl group. Now, this enzyme is going to be regulated by cortisol. So, you've got stress, which is going to increase cortisol, and that cortisol is going to activate or ramp up this enzyme. So when cortisol levels increase due to stress, more epinephrine will be produced. Our epinephrine levels will increase. You may also want to keep in mind that both dopamine and norepinephrine are produced both in the central nervous system and in peripheral tissues. However, epinephrine here is only produced in the medulla of the adrenal glands. So I want to emphasize that. Epinephrine here is only produced in the adrenal medulla. Alright, let's play a quick game here. Now, we've spent all this time to build up our catecholamines. Now, how do we break them down? Remember, I like to build stuff up and break it down at the same time. So let's play a quick game. Catecholamines can be broken down by our Pac-Man enzymes, monoamine oxidase, or MAO, or catechol O-methyltransferase, aka COMT. Now, dopamine is broken down by MAO and COMT into homovinylic acid. Alright, norepinephrine is broken down into VMA, or vanillomandelic acid, and lastly, epinephrine is going to be broken down into metanephrine. Metanephrine here. One disease uses these breakdown products as a measure of catecholamine production. Now, that's going to be a pheochromocytoma. A pheochromocytoma is a tumor of the adrenal medulla chromaffin cells, and that produces high amounts of catecholamines. You can use these lab values here of the breakdown products to help diagnose a pheochromocytoma. Now, this disease is discussed in more detail later on in the organ systems lectures. So, since we are all out of catecholamine coins, we are going to call game over for our catecholamine breakdown.